Dear students, welcome to today's session. We will explore the fundamental biological processes that sustain life and protect us from disease, starting with hematopoiesis, the stepwise generation of blood cells from stem cells, and moving into the intricate defense system of our body, innate and the adaptive immunity. By understanding how these systems develop, function and coordinate, you will gain insight into both normal physiology and the basis of important disorders, as well as how our immune memory forms the foundation for disease prevention through vaccination. Yes, you are right. We are going to discuss about structure and functional aspect of immune organs and cells, focus on hematopoiesis, adaptive and innate immune components. The learning objectives of today's topic are to explain the stages, anatomical sites and cellular pathways of hematopoiesis, to analyze the regulatory mechanism of hematopoiesis, including the roles of key cytokines and growth factors, to differentiate between innate and adaptive immunity in terms of components, mechanisms and functional attributes, to describe the cellular and molecular mechanism of humoral and cell-mediated immune responses, to evaluate the integration of innate and adaptive immune systems in protecting the body against infections and tumors. We will start with hematopoiesis, the process of blood cell formation. Hematopoiesis comes from two Greek words, haima means blood and poiesis, which means to produce, is the lifelong process of generating red blood cells, white blood cells and platelets beginning in the embryo. All blood cells originate from hematopoietic stem cells or the HSCs, unique self-renewing cells capable of differentiating into diverse blood cell types, sustaining oxygen transport, immunity and clotting functions. The sites of hematopoiesis. In embryonic stage, yolk sac is the first site of blood cell formation during the 3rd week of gestation. In fetal stage, liver is the main site of hematopoiesis during the 2nd trimester, and spleen is the second site during fetal development. In adult stage, the bone marrow is the primary site after birth, especially in flat bones, for example, sternum, pelvis or ribs. Hematopoietic stem cells or the HSCs. HSCs are multipotent stem cells capable of self-renewal and differentiation into all blood cell lineages. They reside primarily in the bone marrow and serve as the foundation of the hematopoietic system. Differentiation pathways of hematopoiesis. HSCs give rise to two main progenitor lines, the common myeloid progenitor and the common lymphoid progenitor. The myeloid lineage is derived from common myeloid progenitor. This lineage produces erythrocytes or the RBCs, transport oxygen. The megakaryocyte, which produce platelets for blood clotting. The granulocytes, which include the neutrophils, eosinophils and basophils. The monocytes, which differentiate into the macrophages and dendritic cells. Lymphoid lineage, derived from common lymphoid progenitor. This lineage gives rise to B lymphocytes, which provide humoral immunity and produce antibodies. T lymphocytes provide cell-mediated immunity. Natural killer cells provide innate immune response. Regulation of hematopoiesis. Hematopoiesis is tightly regulated by network of cytokines and growth factors. Key regulators include erythropoietin, which stimulates RBC production, thrombopoietin, which regulates platelet production, granulocyte colony stimulating factor, which promotes granulocyte production, interleukins, which support various stages of lymphocyte development. We will now be discussing about the disorders of hematopoiesis. Anemia, red blood cell deficiency, leukopenia, low WBC count, leukemia, uncontrolled proliferation of abnormal white blood cells, thrombocytopenia, low platelet count. Hematopoiesis is a dynamic and essential process for life, responsible for the continuous generation of all blood cell types. Understanding this process is critical for diagnosing and treating many hematological and immunological disorders. The second part is immunity. Immunology encompasses the physiological processes enabling the body to recognize and respond to foreign materials, neutralizing, eliminating, or metabolizing them ideally without harming its own tissues. The immune system, a complex of networks, cells, tissues, and organs, protects against the infections and diseases. Its hallmark is distinguishing self from non-self, forming the foundation of clinical immunology and guiding the mechanism that recognize and respond to foreign structures in health and disease. First of all, we will be discussing about innate immunity or the non-specific immunity. Innate immunity is the natural protection present from birth, serving the body's first line of defense against infections. It operates through non-specific disease resistance mechanism, active before infection, recognizing common molecular patterns of pathogens. Key components include phagocytic cells such as macrophages, neutrophils, physical barriers such as skin, mucous membranes, antimicrobial compounds, and normal microflora, all working together to prevent pathogen entry and spread. Innate immunity comprises different types of defensive barriers such as anatomical barriers, cellular components, physiologic barriers, phagocytes, inflammatory response, and other innate barriers. First of all, we will be discussing about anatomical barriers. The skin and the mucosal surface, physical and anatomical barriers constitute the body's first line of defense, preventing pathogen entry and providing continuous broad spectrum protection. Second, the skin, comprising the outer epidermis and the inner dermis, blocks most of the microorganisms. The epidermis consists of tightly packed epithelial cells with the keratinized outer layer offering waterproofing, while the dermis contains blood vessels, follicles, and glands. Sebaceous glands secrete sebum rich in lactic and fatty acids, maintaining an acidic pH of 3 to 5 that inhibits microbial growth. Fourth one are the breaches such as wounds or insect bites, which permit the entry of pathogens as in malaria, plague, and the Lyme disease. Mucous membrane linings, the conjunctiva, and the alimentary canal, respiratory, and urogenital tracts further protect by secreting mucus, saliva, and tears containing antimicrobial agents. Fifth one, in the respiratory tract, ciliary actions remove the mucus and trapped microbes, preventing colonization and infection. Now we will be discussing about the cellular components of the innate immunity. These immune cells detect and destroy the pathogens by various ways of phagocytosis, cytotoxic activity, or the secretion of signaling molecules. Phagocytes such as neutrophils, these are the first responders, engulf and destroy pathogens. Second, the macrophages. These phagocytose pathogen and dead cells, present the antigens to the T cells. Third, the dendritic cells. They capture the antigens and initiate the adaptive immune responses, link between the innate and the adaptive immune system. Fourth one, the natural killer or the NK cells. They kill the virus infected and tumor cells by inducing apoptosis. Fifth, the mast cells, which release histamine and other mediators during inflammation and allergic responses. Sixth, the basophils and the eosinophils involved in defense against parasites and allergic reactions. Next are the physiological barriers. Physiologic barriers in innate immunity such as temperature, pH and soluble molecules like lysozymes, interferons and the complement proteins limit pathogen growth by degrading cell walls, inducing antiviral states or damaging membranes, while some species naturally resist diseases because their body temperature prevents pathogen proliferation, collectively offering broad, non-specific protection against infections. Next are the phagocytic barriers. Phagocytosis and innate defense by monocytes, neutrophils and macrophages engulfs extracellular particles like pathogens via plasma membrane extensions, forming phagosomes, enabling effective capture and elimination of harmful microorganisms. Next are the inflammatory response. Inflammation is a protective biological response to harmful stimuli such as pathogens, injury or irritants. It coordinates cellular and molecular events to eliminate the cause, remove damaged tissue and initiate repair. Microbial components like lipopolysaccharides or the LPS trigger the process via specific cell surface receptors. The four cardinal signs, which are the redness, heat, swelling and pain, reflect three main events such as vasodilation of nearby capillaries causing redness and heat. Next is the increased capillary permeability, allowing protein-rich fluid into tissues leading to swelling, and phagocyte influx through margination, diapedesis and chemotaxis to eliminate harmful agents. Phagocytic cells engulf pathogens, but release of lytic enzymes may also damage healthy tissue. Dead cells, digested material and fluid from pus. Chemical mediators regulate the process. Acute phase proteins rise during infection. Histamine induces vasodilation and permeability. Kinins like bradykinin promote vasodilation, permeability and pain. Enhanced permeability also allows blood clotting enzyme to deposit fibrin, forming a barrier that walls off the affected area, prevents the infection spread and initiates the tissue repair. Last is the other innate barriers, the bony encasements. Bony encasements such as the skull and the thoracic cage protect the vital organs from injury and the entry of microbes. Mechanical removal. The process of physically flushing the microbes from the body include the cough and the sneeze reflex, which remove the mucus and trapped microbes. Vomiting and diarrhea. These processes remove the pathogens and toxins in the gastrointestinal tract. The physical flushing action of body fluids. Fluids such as urine, tears, saliva, perspiration and blood from the injured blood vessels also flush the microbes from the body. Bacterial antagonism by human normal flora inhibit the colonization of pathogens by producing metabolic products such as fatty acids, bacteriocins, etc. that inhibit the growth of many pathogens. We will now be discussing about adaptive immunity. It is a specialized defense mechanism that recognizes and eliminates specific foreign microorganisms and antigens. Adaptive immunity displays 4 characteristic attributes: the antigenic specificity, the diversity, the immunologic memory, and the self or non-self recognition. Unlike the innate immunity, it responds to unique antigenic challenges, showing variability among individuals. Its antigenic specificity enables the discrimination between even subtle antigen differences, and its vast diversity allows the recognition of billions of distinct antigenic structures. Once exposed, it develops immunologic memory, producing a stronger, faster response upon re-exposure to same antigen. Importantly, adaptive immunity demonstrates self or non-self recognition, targeting only foreign antigens while sparing the body's own cells, an ability essential for preventing autoimmune reactions and ensuring precise immune protection. There are 2 major branches of adaptive immune responses. The first one is the humoral immunity and the second one is the cell-mediated immunity. We will now be discussing about the humoral immunity. The humoral immune response, a key arm of adaptive immunity, targets extracellular pathogens like bacteria, viruses and toxins in blood and lymph. Mediated by B lymphocytes from the bone marrow, it begins when B cell receptors bind specific antigens. B cells may act as antigen presenting cells via MHC2 and upon activation differentiate into plasma cells secreting antibodies, which are Y-shaped immunoglobulins with antigen binding Fab regions and an FC region for immune interactions. The 5 antibody classes such as IgG, IgM, IgA, IgE and IgD perform distinct protective functions. The process of response. The humoral immune response begins when a B cell receptor called as the BCR binds an antigen. In the T-dependent activation, B cell processes the antigen and present it via MHC2 to activated CD4 T helper cells, which provide co-stimulation and cytokines, driving the clonal selection and differentiation into plasma and memory B cell. T-independent activation occurs when repetitive antigens directly cross-link the BCRs, producing rapid IgM without memory formation. The role of antibodies in combating infection. Antibodies combat pathogens through neutralization, opsonization, complement activation, and antibody-dependent cell-mediated cytotoxicity (ADCC), enabling the pathogen lysis or destruction of the immune cells. These mechanisms ensure precise, immediate, and long-lasting protection against extracellular threats. Immunological memory, a hallmark of humoral immune response, arises when initial antigen exposure generates long-lived memory B cells alongside plasma cells. These memory cells persist in circulation and lymphoid tissue for years. On re-exposure to the same antigen, they rapidly activate and proliferate and differentiate into plasma cells, producing larger amount of higher affinity antibodies than naive B cells. This swift, potent secondary response underlies vaccine effectiveness, as vaccines prime the immune system against future pathogenic encounters. Second is the cell-mediated immunity or the CMI. Cell-mediated immunity is a critical arm of adaptive immune system that protects against the intracellular pathogens, tumors and transplanted tissues. Unlike antibody-mediated responses, CMI relies on antigen-specific cytotoxic T lymphocytes or the CTLs, helper T cells, activated phagocytes and the cytokine release. Adaptive immunity is defined by specificity, precise antigen recognition, heterogeneity, diverse cell types and molecules, and memory, faster, stronger response upon re-exposure. CMI is initiated when antigen presenting cells or the APCs display antigens via MHC molecules to naive T cells, triggering the proliferation and differentiation. CD8 CTLs kill infected or abnormal cells using perforin and granzymes, while CD4 helper T cells release cytokines to enhance the macrophage function, activate CTLs and support the B cell responses. The regulatory T cells limit the excessive immune activity to prevent tissue damage, ensuring immune homeostasis. This coordinated system provides targeted, long-lasting defense against intracellular threats. We will discuss about the main components of cell-mediated immunity. T lymphocytes or the T cells. T cells are the central players in the CMI. They originate in the bone marrow and mature in the thymus. CD8 cytotoxic T cells or the CTLs. CD8 T cells are known as cytotoxic T lymphocytes or the CTLs. The main function is to directly kill the infected or abnormal cells, recognize and kill virus infected, intracellular bacteria infected and tumor cells. Recognition occurs through interaction between the T cell receptors or the TCRs and the antigen MHC class I complex on target cells. Induction of apoptosis. CTLs release perforin and granzymes. Perforin forms pores in the target cell membrane. Granzymes enter through these pores and activate the caspases, triggering the apoptosis. Cytokine secretions. Release of INF gamma or the interferon gamma, which activates the macrophages, enhance antigen presentation and also inhibit viral replication. Immunosurveillance. Patrol the body for abnormal cells such as cancer cells or the virus infected cells and eliminate them as soon as possible. Memory CTLs, after infection is cleared, some CD8 T cells differentiate into memory T cells, providing the long-term protection against re-exposure to the same antigen. CD4 helper T cells or the TH cells. CD4 T cells are essential regulators of the immune system. They do not directly kill the pathogens but assist other immune cells via cytokine secretions. Functions. Functions include the activation of other immune cells, recognize antigen presented on MHC class II molecules by dendritic cells, macrophages, B cells. After activation, they differentiate into various subsets like Th1, Th2, Th17, TFH or the follicular helper T cells and the TREG or the regulatory T cells. First one is the Th1 cells which produce IFN gamma which activates macrophages for killing intracellular pathogens like Mycobacterium tuberculosis, supports CD8 T cell activation. Th2 cells which secretes the interleukin 4, 5 and 13 and help the B cells class switch to produce IgE, recruit eosinophils and basophils important in parasite infections and allergic responses. The Th17 cells produce interleukin 17 and interleukin 22, recruit the neutrophils and enhance the barrier immunity, especially against fungi and extracellular bacteria. The next are the TFH or the T follicular helper cells, localized in the germinal centers of lymphoid organs. They provide the help to B cells for class switching, somatic hypermutation, antibody production. Next are the TREG or the regulatory T cells which express the FOXP3. They secrete interleukin 10 and TGF beta to suppress the immune response, maintain self-tolerance and prevent autoimmunity. The secretion of the cytokines modulates the immune response by tailoring it to the type of pathogen or the antigen encountered. Memory helper T cells persist long term and enable quicker and stronger response on re-exposure to pathogens. Macrophages in the cell-mediated immunity. Macrophages are phagocytic antigen presenting cells or the APCs that originate from the monocytes. They play a central role in both innate and adaptive immunity, particularly in cell-mediated responses. Antigen presentation. Macrophages ingest pathogens, degrade them into peptide fragments and present them on MHC class II molecules. These antigen MHC complexes are recognized by CD4 helper T cells, initiating their activation. They also express co-stimulatory molecules, example CD80 or CD86, needed for full T cell activation. Phagocytosis. Macrophages engulf and digest pathogen like bacteria, viruses, protozoa, dead or infected cells and cell debris. This occurs via pattern recognition receptors or the PRRs that recognize the pathogen associated molecular pattern PAMPs. Cytokine secretion upon activation. Macrophage secretes cytokines like interleukin 1, 6, TNF alpha, promote inflammation. Interleukin 12 promotes Th1 differentiation and activates NK cells. Chemokines attract other immune cells. Tissue repair. Macrophages also help resolve the inflammation and promote tissue remodeling and repair after infection is cleared. Dendritic cells. In cell-mediated immunity, dendritic cells are the most potent antigen-presenting cells and are essential in initiating adaptive immune responses. Antigen capture. Immature dendritic cells reside in the peripheral tissue. They capture the pathogens through phagocytosis, receptor-mediated endocytosis and macropinocytosis. Antigen processing and presentation. Dendritic cells process antigens and present them via MHC class II to CD4 helper T cells, MHC class I through cross presentation to CD8 cytotoxic T cells. This dual presentation capability makes them crucial for initiating both arms of cell-mediated immunity. Cross presentation unique to DCs. They can present extracellular antigens on MHC class I to CD8 T cells, activating cytotoxic T lymphocytes. This is vital for immunity against viruses and tumors. Natural killer cells or the NK cells, natural killer cells, part of innate immunity, eliminate virus infected and tumor cells without sensitization or MHC recognition, aiding cell mediated immunity. They patrol tissues targeting stressed or MHC class I deficient cells. Killing involves perforin mediated pore formation and granzyme induced apoptosis, enabling the rapid, broad defense against abnormal cells. In conclusion, hematopoiesis is the continuous process of generating all blood cells such as red cells, white cells and the platelets from multipotent stem cells in the bone marrow, maintaining the oxygen transport, immune defense and homeostasis. Disruption can cause disorders such as anemia, leukopenia or leukemia. Effective immunity relies on two complementary systems, innate and adaptive. Innate immunity provides rapid, non-specific defense via barriers, phagocytes and soluble mediators, while the adaptive immunity mounts slower, highly specific responses through T and B lymphocytes, producing immunological memory. Their coordinated action ensures immediate protection and long-lasting defense against diverse pathogens. Dear students, this was all about today's lecture. I will see you next time. Thank you.